Welcome class, welcome in the third video lecture of the microscopy. So today I am going to give you the solution of the problem that we have seen in the second video lecture, that was all about bright field microscopy. In bright field microscopy we have seen a problem that we cannot visualize. We cannot visualize any live sample in bright field microscopy, right? That is because of the semi-transparent nature of biological sample, which as a result they lack a contrast with their background, right? And as a result we have to stain them to create the contrast, but unfortunately stains are chemical reagent, so it causes the death of the specimen. Okay, so that was the problem, that we cannot visualize very well a live samples with the help of bright field microscopy. Okay, so now scientist starts to think about this, that is there any way with the help of which we can see a live microbes? Is it possible? So while searching they find out a way that if we lower the amount of light that we are providing in the specimen, that means here we are using a darkness, right? That means we are reducing the amount of light we are providing in case of bright field microscopy. By reducing the amount of illumination, there can be a possible way by which we can see a live microorganism without any staining. Let's see how. So here scientists develop a system where they develop their condenser in such a way that it consists of a specialized size disc named as patch stop. This patch stop prevents all the amount of the light that is transmitted through the specimen. As a result, the illumination becomes lowered. So what is the utility of lowering the illumination? So the utility of lowering the illumination is, let me told you will get something like this. At low light there will be a bright cells you can visualize under the dark background. Okay, you can clearly visualize that yes, it is a cell, and inside the cell, if I zoom this picture, then you can easily visualize that yes, these dark ones are cytoplasms, right? So that means with the help of dark field microscopy, by lowering the illumination, we can generate a clear picture of live cell against black background. That can be possible, right? But it contains still some disadvantages. Those disadvantages are, can you make it out? What is this? Is it some of you will call it as a cell and some of you will call it as a artifacts. So now the question is, what is artifacts? Let me tell you. So artifacts is nothing but a type of aerosols, a type of impurities which is coming in our slide during our slide preparation, and that have the ability to absorb the low amount of the light that we have provided, and they become brightened, and that is why they are also visualized under the bright field microscopy. But some of you may confuse this with the cell. That means we cannot distinguish between what is artifacts and what is cell with the help of dark field microscopy. But on the other hand, when we are using bright field microscopy, we are providing a too much amount of light, right? And under those too much amount of illumination, only the cell will be absorbed, and they become illuminated, although this illumination is with the field, but when we apply stain we can clearly visualize them. But this is the problem with dark field microscopy, that dark field microscopy cannot distinguish what is impurity and what is the live cells. So that is one of the major disadvantage of dark field microscopy. So in dark field microscopy, by lowering the illumination, we are capable of seeing live cells, because here live cells will absorb those illuminated light and they become brightened, that we can visualize. But although they solved the problem, the major drawback of the bright field microscopy, that is we cannot visualizing live microbes with the help of bright field microscopy, that problem is solved by dark field microscopy. But dark field microscopy itself have some problem, that is we cannot distinguish what is cell and what is artifact. Okay, another important problem that I want to mention, that is, yes, you can easily visualize what is the surface architecture, no doubt about this, but can you visualize which one inside is the ribosome, which one inside is the nucleus, which one inside is the mitochondria? That means the internal analysis of the sample cannot be done with the help of dark field microscopy. Okay, that means if you want to only visualize how live cell look like under the microscope, then you can go for dark field microscopy. But for detailed analysis you have to either rely on bright field microscopy, or you have to move on the further advanced microscopic technologies. But dark field microscopy under such circumstances cannot help you. Okay, and another important disadvantage, that most of the image will be only the black and white. Okay, so in dark field microscopy what we are doing, we are lowering the amount of the light we are providing in case of bright field microscopy. By lowering the amount of the light we provide, we are creating a picture that can clearly provide us details of the surface of the live cell. Okay, so the major drawback of the light microscopy is solved, but it cannot distinguish between what is artifact and what is cell. Also it cannot provide us the details of the inside of the cell. So that are the kind of disadvantages of dark field microscopy. But no doubt dark field microscopy is necessary for visualizing the live microscope. Most of it is necessary for visualizing the live cell that our light microscope cannot provide us. So now let's see how it is going on. So the condenser used in light microscopy help us to concentrate and transmit it through the specimen, right? Now this gives a bright field illumination. However, dark field microscopy is more useful in analyzing light sensitive organisms, to examine those specimens that have a very small differences or contrast with their background in a bright field illumination. Okay, so a microscope adapted for dark field illumination is nothing new. It is as same as bright field microscopy, but in addition to bright field microscopy, it has a modified condenser system. This condenser system contains a specialized size disc called patch top just beneath the condenser, and it prevents light from being transmitted through the specimen. Okay, so that is the principle of the dark field microscopy. So let's talk about the advantages. So no doubt it is extremely simple to use, easy to set up, and we can visualize unstained live sample with the help of these. But disadvantages is it can only provide limited color. You can visualize your sample in a black and white in most of the time. An image can be difficult to interpret for those who are not familiar with those kind of dark field microscopy. Although the surface detail is very apparent, but dark field microscopy cannot tell us about the inside of the cell. And the main limitation that I want to told you, that the presence of artifact is hard to distinguish. Okay, so here we are using the light source. This light source is first of all prevented by the patch stop. This patch stop will prevent most of the light from transmitted through the specimen. Only small amount of light is transmitted towards the slide containing the specimen, and it will ultimately goes to the objective lens and then eyepiece lens and provide us something that we have seen in the previous slides, right? So let me wrap up this topic, that in our previous video we have completed our previous video with a problem that bright field microscopy cannot provide us image of live microorganism, because our microorganisms or any kind of biological specimen are semi-transparent in nature. So if we are using too much amount of light, too much amount of illumination, so along with the background, the specimen will also be illuminated, and as a result there will be no contrast in between them, right? That is why for generating the contrast we have to stain them, and for staining it causes the death of the cell. As a result, with the help of light microscopy we do not visualize any kind of live samples, right? So for solving that problem we have developed the dark field microscopy, which is nothing new. Here we are just lowering the amount of the illumination that we are providing in case of light microscopy, okay, with the help of a specialized size disc called patch stop, that is just beneath the condenser. Okay, it will prevent the amount of light transmitted through the specimen, and as a result very small amount of light will be in our eyes, and in presence of those low amount of light the object can absorb those low amount of light, and they become brightened under the dark background, okay, and that can be easily visualizable. That's how dark field microscopy provide us the opportunity to visualize live microorganism or live cells or live biological specimen that cannot be visualized without staining in light microscope, okay. But it as well as provide a disadvantage, that is we cannot distinguish what is our artifact and what is our specimen, okay. So that is another disadvantage, and not only that, the inside of the cell cannot be visualized with the help of this dark field microscopy. We can only study what is in their surface, okay, but inside of the picture cannot be visualized, okay, and it become often difficult for those who are not familiar with the image produced by dark field microscopy. So that was the lecture about dark field microscopy. So here we have faced another problem, that is we cannot visualize the insides, right, of live microorganism, and we cannot also distinguish what is artifact and what is our cell. So in the next video we are going to discuss about the solution of those two problems. What is the microscopic technique that help us to visualize the inside of the live microorganism, and also what is the microscopic technique that help to distinguish us between what is the which ones are artifact and which ones are our sample, that is our live microorganism, okay. So that was the lecture about dark field microscopy. So I hope this video will be helpful to you. Thank you.